Aloha! We're here in Honolulu, Hawaii for the Plant Biology 17 Annual Conference. I'm your host, Rishi Masalia, and we're Between the Palms. Hi, and welcome to another episode of Between the Palms. Today I am joined by Barnett Rosenberg, Professor of Plant Biochem at MSU, Rob Last. Rob, thank you so much for being on the show.
Pleasure.
Can you tell us a little bit about the research you're doing at Michigan State?
Yeah, so my lab is interested in how plants use biochemistry, metabolism, both to produce molecules of importance to them, that is protective, for example, antioxidants or anti-insect compounds, but also in some cases, such as amino acids and vitamins and things like that, are nutritionally important to us as consumers of.
So you study the production of those.
That's right.
Secondary metabolites.
And how those pathways have evolved over time. Yeah.
Nice. So one of the questions I love to ask plant biologists is, what are your origin stories? How did you get started in plant biology?
From synthetic organic chemistry, and I bet you expected that as an answer, right?
From a scientist, absolutely.
As an undergraduate, I was very privileged to be able to go to a small liberal arts college.
Okay. Where'd you go?
To Ohio Wesleyan University in
Okay.
Delaware, Ohio. And I was really lucky because in the 70s, they still do, I believe, they had a very strong organismal biology program, but also a really strong chemistry department.
Okay, so you got both of those worlds right off the bat.
Nearly a double major with nearly. Well, one course shy of a
credits
double major.
Okay. I'm glad credits are still finicky now as they were back then.
They are. And so I got to learn about organisms, which has served me well. And I got to learn about organic chemistry, analytical chemistry, and so on, and that's what we do for a living, studying those organisms. I thought I was going to go and work on enzyme evolution, protein evolution, but that was impossible back then because the only proteins that we had sequenced for were highly conserved proteins, not very interesting studying evolution. So 40 years later, we're studying protein evolution.
So you finally made it over.
That's right. I made a transition into doing yeast genetics, studying RNA splicing before.
We're at a plant conference.
Yeah, I know. No, I was studying. This was a few years ago.
Okay.
So I went to Carnegie Mellon University, worked for John Wolford, and learned how to do genetics using yeast. Came back to biochemistry, working at Whitehead Institute for Biomedical Research, more cognitive dissonance for you there, working in Gerald Fink's group, a very amazing yeast and other genetics. So I was involved in the short time that his lab did Arabidopsis research, and that's how I got involved back in plants.
Do you work in your lab at MSU, do you work on Arabidopsis or do you work on other?
Yeah, Arabidopsis is waning in my group. I still have two really great postdocs.
Still sticking to Arabidopsis?
Yeah, for what it does, we use Arabidopsis for the kind of metabolism that many plants do, or all plants do. So amino acid metabolism, and we have a bit of an interest in photosystem 2 repair.
So kind of the core.
Core functions. Yeah. Old. You think about them as sort of old biochemistry, old metabolism. Very important, but old. I guess as you get old, you don't necessarily become less important. But we use tomato and its relatives in the Solanaceae to study specialized metabolism. So clade-specific, often tissue-specific metabolic networks that make sticky stuff, for example, on the leaves of petunia and tobacco. And in our case, we start with tomato because of the wonderful genetic tools and genomics tools. We do our discoveries, and then we've been working out evolutionarily. We're at about 50 or 60 million years. So now studying enzyme evolution, protein
Nice.
evolution.
So when you had mentioned that metabolisms can be used for benefits for humans as
Yeah.
well as defensive compounds against herbivores.
Yeah.
So do you have a preference? Do you study both in your lab?
Well, so the compounds that we're studying right now are they're used industrially as emulsifiers. I think that they have a role in tobacco taste, but I'm not so interested. I mean, I'm not so interested in that. We're interested in it from a basic biology perspective because they evolve very fast.
Oh,
okay. I mean,
nice.
you know, we see intraspecific variation in these phenotypes of the metabolites and the underlying enzymes. That's evolved over hundreds of thousands to early millions of years. So that's a big reason why we're interested, because I'm really
Just this foundational, foundational work.
There are people elsewhere in the world, Martha Mutchler at Cornell, for example, doing breeding, trying to increase the amounts of these acylated sugars, they're called, in cultivated tomato. And we collaborate.
I was going to ask
and
you guys collaborate.
Yeah, that's right. So when we find new enzymes, we tell her about them.
You're saying so you don't just stick in your own little bubble and tell nobody about it?
We like to joke in my lab that every time we publish a paper, of course, we're very excited about it, and we hope that at least 5 other labs around the world are excited. So I think hopefully it's at least 10.
At least
10.
Hopefully.
Yeah.
So you were president-elect of ASPB, and you were also one of the founding members of the ASPB Legacy Society.
That's right.
Can you tell us a little bit more about that?
The Legacy Society was established by ASPB this last year, really spearheaded by Brian Larkins, a truly wonderful longtime member and former president of the ASPB, probably when it was ASPP, Plant Physiology, though he could correct me on that, where around 70 or 80 of us came forward, prompted by Brian, came forward with contributions to establish what will hopefully be a continued tradition of giving, both for, in this case, endowment, to allow good works over the next 100 years. We've almost made it to almost made it to 100 years. The next 100 years, as well as gifts for operating expenses. To help fund more summer undergraduate research fellowships, or other travel awards and so on. Yeah,
very nice.
That's right. Of course, there's a long tradition of giving at ASPB, but this is
a little bit more focused and continue forward. Right?
Yeah, trying to enervate it. I think that might be a reasonable way of saying it.
Energize it.
Yeah.
Perfect. So one of the things that I know about you is that you have bounced around. Some would say the term you're a polymath. Some would say.
That's Crispin.
Crispin says that. Yeah. So
I had to look it up to see. He's so erudite. I had to go and look up the word when he first accused me of that. Yeah.
So you have worked in academia, you've worked in industry, and you've worked at NSF.
That's right.
And now you're back in academia.
Yeah, that's right.
Can you tell us a little bit about the differences between all 3 of those?
How long do you have? How much tape do you have? I mean, there are huge differences. I mean, clearly within any one of those areas, right? There's a huge range from, for example, being an educator at a primarily undergraduate institution. If we're just talking about post high school, right? From being a PUI professor, a place like where I did my initial training, all the way to research intensive with lots in between, and that's not the full span. Actually, community colleges and so on. Within industry, there's this wonderful range from startups where there are 5 employees and each of them has 6 jobs, right? And everyone is trying to raise funds and stuff.
As opposed to a big company where you have little facets of everybody doing
different things and opportunities to do that kind of career advancement over years instead of days. I happen to have been a director at Syrian Genomics, so a genomics company, a wholly owned subsidiary of Monsanto. So I got involved in industry, not in like ground floor.
Right,
so my experience is very different,
different
from what others might have.
So when you were bouncing around to these different locations over the course of your career, what made you decide to transition from one to the next?
Oh, opportunity, necessity. But so I'll just give you an example of opportunity, right? In 1997, 1998, there were not many places where you could do genomics. Yeah, sure, you could sequence, your gene and blah blah blah, screen a back library, things like that. But full-fledged, what we now think of as just what you do, which is like we used to call it EST sequencing,
RNA-seq now, you gotta get everything.
Genome sequencing and so forth, just wasn't really available in places other than a few ahead of the curve elite private institutions on the coast. So I got this opportunity to help found this genomics company and do some really cool stuff. So, guess what? We actually sequenced the Arabidopsis genome first. We did about a 1.1, 1.2x coverage of Landsberg erecta to complement what was being done in the public sector on Columbia, and then we released the polymorphisms, and people have been using them ever since, right? Nice. Just as an example. Yeah.
So what is it like you worked at NSF?
For a year and a half.
Yeah, so what were you there?
Program officer, primarily in plant genome research.
So can you give us a little bit of a description of what you were doing as a program officer?
You read grant proposals, you help find reviewers for them, you help organize or collaboratively organize panels, right? So review panels. You read annual reports and give feedback to the PIs. One of the things that I did right at the end, so Machi Dilworth was my boss, and Machi was always encouraging in so many ways, but one of the ways was that she kept saying, Rob, you really need to go to Capitol Hill and go to some some hearings and see what it's like. Towards the end, I did go to an Senate appropriation committee meeting, and it was, hearing, like the opening one for that cycle, and it was really cool. It was back when when politicians seem to like each other. And so, for example, watching Kit Bond and Barbara Mikulski interact together, at that point, maybe he was chair and she was ranking member, or vice versa, and then interact with the NSF staff leadership who were there, in a friendly and constructive sort of way. It was very cool.
That's cool that you gotta see the process of helping other faculty members achieve grants, get grants and funding, and then also watching the policy side as well.
Yeah, even being able to do some mentoring by phone.
Really?
Shinhan Shu, who's now a good friend and a colleague at Michigan State. First time I met Shinhan was on the telephone. He called. There's this guy, speaking with this Taiwanese accent, asking me for advice, and we ended up talking for an hour or so, and
now you guys are
colleagues
at MSU.
We go out for beers and hot pot together on a regular basis.
Well, that sounds great.
So you even have the opportunity to have a positive impact on, everybody thinks about, my grant won't get funded. Well, unfortunately, that does happen.
That does happen, but there's other benefits, right?
Yeah.
Aside from that.
Which is cool, yeah.
Very good. All right, well, I think that's going to do it for this episode of Between the Palms. Rob, thank you so much for taking the time.
Pleasure.
And we'll see you all next time.